I put all these together in a presentation, so probably it's a little bit messy, but well, I thought that it was worth to share with everybody. So I work in the past a lot with microRNAs. Um, so for some reason, I tend to still be attached to that field. I don't know what to do. Let's see. Let's see. Okay. I I don't know if I'm sharing anything or not. Somebody can talk to me.
There, you're back now, Lorena.
Okay. Sorry, I don't know what happened. But anyway, so I will talk about small RNA sequence data analysis. So I just will introduce a little bit microRNA and isomers. Well, I'm sure almost everybody knows a little bit about them. I just going to as well put a couple of slides about the protocol, the sequencing protocols, because like normally, like I will say 90% of the people will use Illumina, but now there is this other bioscientific that the idea of that protocol was to ligation by then like everybody normally has a way to map microRNA, and so there are different ways to do it. So I try just to put all together what people, well, I have read in methods in paper do, and see more or less if there is a better solution or not for that. And as well, there are few people, although I'm seeing in papers and publication that isomers start to be like more the target of the study. So I will talk about what is the main problem of isomer annotation, or what people do, and maybe a possible solution so we can all do the same together, kind of. And then I will just end up with what we do in BCBio or in our bioinformatics core to analyze this isomer microRNA using an R packet. So it will be more or less what I normally do when I have this kind of data. So yeah, microRNA, very small molecules. The main function of the small RNA, microRNA are regulation. So they bind through other proteins to the mRNA, and then this mRNA can go to degradation, or they just stay sequestered there in some protein complex. So they are very important for therapeutical studies. My mid connection is unstable. So, but beside microRNA, because people, like many people, just think about microRNA like a unique sequence, that is the one that has the main function. But what happened is, like when you sequence the small RNA of a sample, you will see variation. So the precursor that is in the bottom of the slide normally generate two kind of microRNAs that are in this pink color. And when you sequence small RNA of any sample, you will see that beside this specific pink sequence, you see a small variation around that. Like normally, like 1, 2, 3 nucleotide shifting, or some mutation for sure that some of them are error sequencing, and as well some nucleotide addition. So this is something that at the beginning was like, oh, it's error of sequencing and all this stuff, but actually people have proven that they are there. They load to AGO, that is one of the protein, to the functional regulation that they do. and even they put it in the cell and they tested that they are doing things. So I think that it's important that people know about them and maybe they decide what to do with that information. So type of isomer. So we can define like mainly 4 types, and everybody call them differently, for sure, because there is no standard pipeline for that or nomenclature. So for sure you have the nucleotide substitution. That is what you find in every other type of data. But then you have changes at the beginning and at the end. So at the beginning, you can call these 5' isomers due to 5' trimming events. And what could happen is like if you compare to mirbase, that is the most used database for microRNA, you will see that sometimes the sequences start before the one that you have in mirbase or after. And these are called 5' trimming isomers events. The same happen at the 3' end, so you have this shifting, this trimming changes. And then they are called 3' isomers, trimming events, or whatever people want to call them. And the last one, it will be addition. So the addition are the one, the nucleotides that you don't find in the precursor. So normally, mainly they are adenosine and uracil. I think that if you study this in the samples, normally you find 1, 2, up to 3. More than that, adenosines or uracil after that is weird to see. And as well, there are papers trying to study that. Mainly, it seems that they add stability when they bind to the mRNA in general, and they change with time in different situations. So it seems that for some reason these additions help or are important in some cases. Talking about the sequencing protocol, so the one that is called fixed adapter sequence is normally all these versions of Illumina protocols that mainly you add the adapters at the beginning and at the end, and that's all. So what happened with that is that it's true, I mean, this has been proven, that you have this ligation bias. And sometimes, so that means that the number of counts that you see in a sample, if you want to compare one microRNA or one sequence with other, this value maybe is not representative. So sometimes you can have 1,000 of one microRNA and of other 200, but that doesn't mean that one is more expressed than the other, always, because there is this ligation bias depending on what nucleotide are at the beginning. So what Bioscientific did is change a little bit this protocol, and the main difference is the scheme that you see on the right. That is adapt 4 nucleotide, random nucleotide at the beginning and at the end between the adapters. And that seems to, like the idea is to try to remove as much as possible this bias in the ligation. So the only extra step that you have to do in the bioinformatics pipeline is that after removing the 3' adapter, you have to remove 4 nucleotides at the beginning and at the end. And that should be enough. You suppose that is quite stable, these 4 nucleotides, so it always will be 4. So I only have experience with 1 data set or 2 of this protocol. It's difficult to compare because I don't have what happened if you do the other one, but there is a paper. They almost did all the data available in the Geo database. I think that they missed 1 just by error. But the ones that has the 2 protocols and with the same sequencing technology, HiSeq, is a mouse liver sample. And they have that in the figure. What's on the left, the paper figure, is that the comparison. So in the x-axis, you have the bioscientific protocol. In the other, you have the TrueSeq from Illumina. And they use the pipeline, quantify. Well, I mean, for me, it's not that super bad. The correlation, I don't know. I mean, 0.03 seems too small for that figure. As well, there is a kind of weird filtering in the lower, like if they just remove everything, maybe with whatever counts, because it's normally what it happens when you do that. And only happened in this sample. They have other because they compare other stuff, and so I don't know what happened in this case. But anyway, I download these two samples, and I just ran BCBio just to look at the correlation, and it's the same. So NextFlex is the bioscientific pipeline protocol, sorry, and then TrueSeq. And I mean the tendency is kind of the same, and you have these all the lower points that is supposed to have different expression in the bioscientific compared to TrueSeq, that I guess that these are the ones that is supposed to have a better estimation of the real expression. But then I have this line on the left that is that there are 0 counts for the bioscientific protocol. So I mean, they are like 50 or so, or 70 microRNAs. And I thought that it was an error, something happened. So I went to check like 3 or 4 of them in the FastIQ, like before trimming or anything. And actually they are not there. I mean, any similar sequence to that are there. So for sure, this is only one sample. So I don't know what happened if you do more things, because maybe, I mean, it was different day and technician, or even if it was the same RNA.
These are classical microRNAs.
Yeah.
Yeah. That are just there.
Yeah, they're normal. Yeah, one of them. I mean, they are not these weird rare microRNA.
They're detected nicely intrusive.
Yeah.
And they're not.
Yeah, like 2,000 counts. So yeah. So I think that it's. I mean, I think that it's good to have two, and I think that for very, very low concentration, it's supposed to perform better. This is what they say. But so it would be nice to have like people try to do pilot experiments before just deciding one or another, just to make sure what happened. Because in the bioscientific, they don't do this band selection as normally you have to do with TrueSeq. They use like some magnetic bits that depend on the concentration that you put. You are selecting by some size. So as well, like other different that normally, like I saw, it was super interesting. It's like, since with TrueSeq you do this band selection, that you are picking a little bit longer small RNA, like 33 nucleotide long sequence that normally are tRNAs, and with this one, I guess that if you need to set up this very well, the way that they are purifying small RNA, because you lose that. And there are people that are starting to be very interested in these smaller tRNA fragments. So this is something that it seems that the bioscientific protocol, as it is super focused in purifying some way these 22 nucleotide long sequences. So just information that I found interesting, because people are starting to use it, and I think that it's good that everybody knows what are the advantages or not of that. So microRNA mapping. So normally you have the FastIQ files. In general, like almost everybody collapse unique different sequences, meaning like instead, if you have 1 million, probably when you collapse, you have 55,000 different sequences. And normally there is kind of a standard here. There is this way of notation that you have this X and then the count of this sequence, and in the middle an ID, that could be a number or whatever. So you reduce a lot the number of sequences to map, for sure, because it's not that complex, a small RNA sample. And then normally people or map to the microRNA mature directly, or map to the precursor. And then there is a step that is parsing. So everybody is doing something and calling these different ways and selecting whatever naming they are doing. So what I did, because actually I think that I never saw in a paper, even any of mine related to microRNA. So it was to do the simulation of sequences of these microRNA isomers, and it's only this. I mean, I'm not even simulating the expression or anything, because that will be a step further. It's just how well we can detect this diversity that we find in the when we sequence. And it was not a large set; it's only the 16,000. Normally, you can find 30-something thousand different sequences that map to microRNA in some way or another. But I think that is enough just to know how the tools are performing. So I use several different tools that I will show later. And then I try to compare if the sequence were mapped or not and were mapped to the correct precursor. Here I have to say that I didn't, like sometimes I actually generate a mutation or an addition, and for some reason actually map better to another microRNA of the same family, because that sometimes happens. So there is, it wouldn't be a 1% perfect result, because there are some noise in the simulation as it is now. But it's enough just to see if they are okay or not. So normally people use like normal high throughput read mapper tools like Voltai, Star. At the beginning, Megablast was used, Nova Lightner, then all these Razer suite that they actually perform quite well, although the time is a little bit higher. But and then there are others that are super focused in microRNA, like we're using myaligner, that is a Java tool, and it's a very simple way to map microRNAs. And then the other one that people use as well is as RNA bench. And I found MirExpress, even if it's not up to date, but I found in a paper last year that they use it. So it's like, well, people are still using, so I will try. So these are the ones that are mapped or not. So in general, like more or less everything map the majority there. So I would say that all the Voltai, Voltai 2, BWA, GEM, STAR, all those that are used for other data perform quite well. And then we have others that, well, no alignment. I mean, I tried to set up to get the best alignment, but I didn't spend like a lot of time, like more than one day, trying to tune the parameters to get the best possible alignment. But I spent some, like at least a couple of hours with each of them to try to get the best of them. So Novo, I will say that is the one that maybe, I mean, since you have other things, I will use other. MirExpress, what happened is that is mainly focused in the mature microRNAs. I guess that is losing a lot of isomers. Anyway, it's not super up to date, so I think that that is performing as good enough to be the time that they develop it. And then Razer 3 was the one with more or less more mapped one. So and it's the one that was slower, but slower meaning maybe 20 minutes. So I mean, it's something that you can do if that is the time for a small RNA microRNA mapping detection. I think that you can do it. You want to get them the best of them. I tried to see if any of the isomer changes were causing that the tool was not mapping some of them. So like if you only focus on the left, that's say no. They are the non-mapped reads. And so red will be the ones with additional mutations in the sequence. The blue will be addition only, and the green will be only mutations, and the other color is nothing. So it's just trimming, like shifting. So I'm not sure exactly why you shouldn't detect that. But like in MirExpress, you see that it's mainly these additions that are the ones that are doing that is failing to map. So that's if not, you will get all of the sequences. And in general, there was not more enrichment than that. MicroRacer maybe. And Megablast, it was a little bit of everything. So beside that, any other tool that you choose should get you more or less the truth.
Question: did you do anything to look at? So this is all sensitivity testing.
Yeah.
Did you do anything to look at specificity?
Yeah, so I tried to look like, I count like how many were mapping to the correct microRNA or precursor in this class, because I wanted to be like super, like, let's see if at least at the precursor level they get annotated, because this, you use the precursor to map almost always this, and then the passing step is the one that you use to select what microRNA. But in general, again, I mean, of course, the numbers decrease a little bit because sometimes they map to other places, they don't found the specific one. But in general, again, I would say raster3 works well, Miraliner works well. With Miraliner, you will miss indels, but people is not focusing indels for now, but I think that is something that maybe will be interesting. But the star works well, BWA as well, get you a lot of true positives. So again,
did you look over splice junctions? Like, are they spliced?
Normally you don't have this in microRNA because they are the precursor is very small, and they are these are like 22 nucleotide sequences.
I mean,
Yeah.
they're the same.
Yeah. I don't know of any microRNA precursor that is in the middle of unsplicing element. But for other small RNA, maybe we could be too. So I didn't say, but I use MIR-base, the precursor in MIR-base to do this analysis, the 21 version.
So an interesting data set you might try is there are RNAs, there's much small RNA databases from AGO2 knockouts.
Mm-hmm.
So that they do have some microRNAs that are intron microRNAs that use very alternative pathways, but most of the microRNAs are basically dead in those cells because they lack the ability to create them.
Mm-hmm. Yeah.
And that might give you kind of a more of a real world test. And one of the concerns is that something like Razor 3, it might be being very greedy.
Yeah, that's for sure.
To try to map, you know, if you give it other garbage in there,
Yeah.
Yeah. It may have.
Yeah, that's for sure. And I don't have it here, but I tried some of them with real data sets, even if it's not wasn't IGO, and things change a little. But the idea of that is like nobody did even the simplest thing, that is try to to map things. But for sure, I mean, this would be the kind of perfect scenario, and for sure it will go. So you can send me if you know.
They're public. I mean, they're just look for AGO2 knockouts.
Okay. So I will look. Okay.
Argonaut.
Okay.
But it's like I said, it's not perfect because there are several microRNAs that survive.
Yeah.
But the large majority of microRNAs are completely
nice.
Argonaut dependent.
Yeah.
So they go away.
Yeah, that's good. So that's something that definitely will help as well to see what happens. So I will look for that. So, but then you have another, because this is only like, okay, you map to some precursors or whatever. So what do you with isomers? Because isomers are variation of the microRNA, and everybody is or naming this differently or not naming and just putting the sequence there. And there was actually a review last year talking about this problem, that there is no consensus here. And we tried to meet like a couple of people and say, okay, let's try to do something for this, even if nobody uses it, but well, maybe somebody that want to use it, it will be there. So the idea is to name isomers based in two rules. So one rule will be the type of change that it has, that could be mismatch, addition, trimming, 5' or 3'. And then one guy got the idea to have uppercases for addition. Addition meaning like it's not in the microRNA mature sequence, and lowercase for deletion. So an example would be you put the microRNA name, and then you put 4 different tags for each of the different changes. And then you can, for now it's 0 or NA, we still are working on that, but so 0 will mean anything. that there is nothing, so it will be the same that the mature sequence in mir base. And then for trimming at 5' you have 2 capital nucleotide, that is just meaning that they start before the mature microRNA annotated. And then in the 3' you have a lower T. That means that N1 nucleotide before the mature microRNA. So it's just that there is a shifting there. So this is not like, I mean, we are working on that. This was the first, after some discussion, the first idea we got that could at least give you a quick side about what is that sequence. And this is what, so we just create a project in GitHub, and we use issues to discuss, and we create just a tool in Python that is called this mirtop, whatever. That what is the, I don't know if I here, yeah. So what it does is just take BAM files. So you can use any mapper you want, and it will take BAM files with sequence mapped to precursor, and it will generate an output with a tabular output file giving you all this information. So what I did is like, okay, let's try to compare if we use this this isomer annotation first, if we are parsing well the BAM file, and then to see if we improve a little bit the ratio between true positive versus false negative parsing the BAM file, because you have to make some selections, right? Decisions when you are parsing to select what is the best hit. So it's kind of easy to use. Just you put the species, and then you use the hairpin FASTA file from mirbase, and the structural of the microRNA as well is a file from mirbase. And then you give the SAM file of the BAM file and the output folder, and you can put a lot of BAM files. So it will just put everything together. That was with the idea that you can put different samples. So you do this for only one for all the samples, and it will create you the count matrix and all the annotations in one step. And this is, I mean, it's super quick because it's only parsing, so it's not doing any super computation. So I took the ones that use the precursor to map the microRNA, and then just run the mirtop. And this will be the same because at the end comes from the BAM file, so it's kind of the same thing. The isomer accuracy as well will be quite correlated as before, but it gives you a little bit more information because it's mapping exactly where the sequence was simulated from. So in general, more or less all are doing quite good. Again, Racer 3 and BWA, maybe Star, are working the best of them, but it's not like a big difference except for no aligner. But we know that. So I try to do this ratio between, well, false negative versus true positive. And the first graph is the one just for the first analysis, that is using the BAM file and see the accuracy of the precursor. And the other one is the isomer level or microRNA level. So at least you have, so this ratio is lower if you actually parse in a way, in the best way, the BAM file. So you are getting more true positive versus false negative. But I mean, at the end, it's kind of the same. So the idea of this tool is just to give you some consensus annotation and help you. When you give this data to people, it's easy for them to see how different this sequence was from the microRNA that everybody knows. And one thing that is useful, and I found useful, is like the validation of the PCR, because people then validate that. And isomers are super useful to give the people what sequence they have to use for validation, because sometimes when you do the differential expression, the mature microRNA is not the one like the one in their base, is not the one that is the most differently expressed in your analysis, and it's other with some changes, some other isomers. So they can go directly and say, oh, this is a sequence that I can use for validation. And that helps a lot to them because sometimes what they use is like, oh, this is not validated, the difference is not that big. And it's like, well, yeah, you need to use the correct sequence to, if you do this analysis like it is. So what we use in the core is like, since we are kind of in a pipeline, it's like from the output of mirtop, we plug the files into the isomers R package, so it can treat that directly. We use that because first it's like, actually there are people that only want microRNA differential expression, like forget about all these isomers, put it all together and do the differential expression. There are other people that are starting to work more on that, and it's like, no, I want all the isomers, or I want only a few, like one type of isomers. So with since you have already in the output of mirtop all the information, with this package you can just select what to analyze, or even do multiple things for this. It has a few figures that maybe like people like to see for isomer, general isomer statistical numbers, like the characterization of what you have in the data. Have some way to query the data, like if somebody asks you, oh, this microRNA has this, or when you look at the, if it's differential expressed, it's like how many sequences you have in this microRNA, so you can query the data. And this is something that I use for one of the projects that is a kind of a PCA, but is supervised, so you actually are giving the groups. And the idea is to, if they separate nicely, you can get the top one microRNAs that are helping to that separation. So it's a little bit more automatized than PCA, and then the one that give more information in the principal component, and actually use other kind of statistical. But this at the end is mainly that. So the input is kind of like that. This will be the output of mirtop. It's missing the last column, but here you have all the information that you will need for this. The processing, so how you load that is very similar to any other sequencing data, like a list of files and a data frame for the experimental design. So you will have all the files, sample 1, 2, in the first variable. And in the second one, you just put all the information you want from this file. The only condition is that the order should be the same. So sample 1 will be the first row, sample 2, the second one. And that's important because then you want to annotate them as they are. And this is what I meant when saying that you can generate different Cohn matrices. Like for instance, the first one, it will be microRNA, the 3' and 5' microRNA all together. The other way is like saying, okay, just separate this data into 3' isomers and additions, and everything else you can put it together, because I don't care about the 3' isomers or mutations, because I don't know how much I can trust mutation. So you will have a Cohn matrix only with all different 5' isomers and additions for each of the microRNA, and do the differential expression on that, or anything you want. You have some easy, like quick isomers figures that actually I showed in the last year presentation. That mainly is trying to give you information about where are your isomers in general in the whole samples. So here is the 5' isomer, and then the x-axis is the position respect to the mature microRNA mir base. So normally all changes happen at -1 and 1. And then you have for sure, like the y-axis is number of unique sequence, and the size of the dot is the abundance. So if you are high and it's super big, it's like all the microRNAs are having this kind of isomers. If suddenly you have something in the lower bottom, but it's super big, normally this is just because there are few sequences with that kind of isomer that are highly expressed, but only happen in some of them. And normally you don't see that because normally you see a general tendency in the whole samples. And sometimes this change. Sometimes I have seen this. I have seen more in 1 than in -1. So I don't know. I guess that need to be studied further. But they give you an idea. You can do this for all the changes. And then there's a paper that I have to review, but they have to do some changes to get acceptance. So I was curious because this problem about the ligand bias is something that we all know in microRNA. So they are trying to use a more complex modeling to try to correct for the PCR amplification bias and the ligase bias. So they have many things, but one thing is kind of figure that they have the same sample and they dilute the sample and sequence, just to show that you have different noises at different concentration of the microRNA. And then they use as the correction as we all do for RNA-seq, like the deep sequencing correction, but still you have noise. And if you add factors for the PCR amplification and ligase bias, you get a nicer estimation of the expression. The only problem with that is like for the ligase bias, you need to actually have like a pilot experiment to know exactly, to learn from that. So it's not like everybody is going to do that for each of the experiment, but you can borrow from other that may be similar, and it will kind of work as well. But they didn't went further. They didn't apply this to differential expression or anything. So this is what the editor asked for, and the reviewers, we asked like, well, I mean, what do you with corrected numbers? It's like, then you have to do something else.
The first correction you talked about that you do, which one?
The first correction will be just the library size correction. Instead of using the negative binomial distribution, is using this complex quadratic linear regression. But the first one is only using this factor, that is the library size. That is what you will get from what you do for any other data. So I don't know, but this, I think that is interesting that people are trying to solve this, because I think that is a bias that affects for some reason more to microRNA than any other. And for the differential expression, I just can say what I use. Normally I choose DSIC2 because work good enough. I think that Lima-Boom will work as well good, and if you have a lot of samples, it will be much faster than DSIC2. And then what I sometimes do, because there are a lot of noise when the microRNA is low expressed, like some samples get like super noisy, is just to filter by group. So meaning, like people do is like give microRNA that are expressing, I don't know how many samples, okay. But sometimes I do this, the same rule, but only by group. So in any group, there are 80% of more expressed, just take it. Because sometimes what happens is like, is expressing one sample in one group, in other, and you keep it, and you are into, this is noise that maybe it will decrease a little bit the power, but it's not a special rule or anything. I don't do this a lot, just with when I have enough samples and more than 2 or 3 groups. And this is the example of the supervised clustering that actually this is a PCA. I mean, this is a different principal component of the PCA. The only good thing is that you do this in a few steps, and then you can get directly the top one with a score of how much they are affecting this separation, for instance, that you see in the first, the second graph on the left. And then it's another way to select microRNA if you want, with beside the differential expression one. I guess that if you have more than 2 groups, probably will be more useful because then you can start see better structure. But for the project that they did, it helped, and some of them were validated. So for sure you will get some of them in differential expression as well. So it's not like this is magic, but it's another way to do clustering. And the last thing that you can do with this package is if you are lucky enough to get a project where you have gene expression and microRNA for the same samples, for sure, I mean, with 2 points, it will work. If you have more than 2 points, it will be even better. I had this twice, so I felt like pretty lucky to analyze this data. You can do pattern, like, well, you do the differential expression, you select your genes and microRNA, that's things that are the most important. You do like profile discovery. All this is done automatically, well, in the function, but is that so you will have different patterns for the gene expression, the microRNA, and then if you use, for instance, TargetScan or any other database to pair microRNA with genes, like what is the prediction, and you use the rule of negative regulation, meaning like the microRNA should regulate the mRNA, you can try to pair what microRNA are regulating what gene. And then you can do go enrichment or K-enrichment or anything. And the idea is to put all this together in a figure that it can help you if we give you some information, like there are 3 panels, the right panel on the top, the left panel at the top, it will be all the GO terms or KEGG term or whatever you want to do. And then you will have the number of genes in the figure that are in this term. And you can know which profile they are following. Like for instance, 1, it will be this upregulation along the time. And then in the left, in the right panel, you will have which microRNA are targeting the genes in this term. So if the idea of that was because they were interested in one GO term, and it's like, oh, but how many genes I have there, and how many microRNA are targeting that, and they wanted to know the profile. So it's like I tried to put all this together in the package in a function. But for sure, you have to have all this information. Like, well, mainly you have to have gene expression data as well from the same kind of samples that you have for microRNA. So as conclusion for this, or what I tried to share, was to decide wisely about the protocol to use. And right now, for me, that will be to do if you are not sure about to use the bioscientific or the TruSeq, just do a pilot with 2 samples and see what are the difference. Because it's a pity just to run the whole samples and then, oh, I'm missing this or whatever, right? So I think that the parsing step of the microRNA and isomer annotation is important. And we are trying to use with this project that we have in GitHub and MIRTOP tool to try to solve that. And if some of you start using that and have more idea, please go there. I mean, we told just in the issue page, and we just want to have some kind of a standard. I feel like it's not like I want to impose this is the right way. I would like to have all people say, oh, this is useful and this will be what I need, and then share this with the rest to have something in common, because I think that if everybody has something in common, then we can move forward and not get stuck in this step that is isomer naming problem. I think that the package for isomer and microRNA helps to try to decide how to create a count matrix in a way that is quite easy. So if you are interested in more than microRNA, this can help you. And then DSEQ2, but you can use, I mean, Limabun work. I use a couple of times work as well. So I think right now they are all pretty consistent. And then for sure, if you can get gene expression and microRNA, it helps a lot to the people to focus in what they want to validate, because sometimes they come with a microRNA and they say, oh yeah, I want to do pathway analysis. And you're like, what? Okay, you are really full of false positive and noise because you don't have anything else. So I try to convince people that even if you do array and it's not sequencing, but something to have more information about your data. So yeah, I think that it's the end finally. Yeah, it's mainly that. So if there is any question, I can try to answer.
So what fraction of mature microRNAs have a sequence other than the canonical microRNA sequence?
Oh, all of them. I mean, normally, so the diversity in terms of just number of sequences, you have, how many mature microRNA are in human, like 1,000 something, right? Normally in a sample, you get 8, if it's with well coverage high, you have 800 mature microRNA. But if you look at all the different possibilities, you will see 55,000 different sequences mapping to this 1,800 canonical mature microRNA. For sure, what I found is normally 3-4 of these isomers are equally or much higher expressed than the mature canonical sequence. So I would say with an expression that maybe you feel like you can trust, I would say 5 or 6 more times than the canonical sequence, as possible other sequences that maybe are doing something else for the diversity. Is that good?
How do you recommend scaling? You talked about that in 1 or 2 slides, normalization.
The normalization.
Between RNAs and microRNAs. I think that shorter sequences by amplifying more.
Yeah, I mean, the PCR amplification is something difficult to fix because what happened with microRNA that you sequence always, all the time, the same sequence. So with gene expression, you can assess that because you will have different fragments along the gene expression. So if you have some bias in some regions, maybe you can fix it with the other regions that don't have this bias and estimate better. But with microRNA, always is the same thing. So it's difficult to know if this is the real expression or is a bias. So it probably is a mix of the two things. So, and this is what the paper that is under review is trying to correct for. But to correct for that, you need to know what you put in the sequences. So you need some kind of pilot experiment every time that you do something super different, and put all these diversity that you think you have and sequence, and then you estimate the factor, and then you can use this factor to many other experiments. Or this bioscientific new protocol, they are trying to fix this PCR amplification as well and the bias ligation. So for now, it's not a way to fix that in a sense that you can get the true estimate, the true value of the expression. The good thing is that more or less should be affected, at least the bias ligation, since it's the same sequence, it should be affected for all the samples the same, more or less. So we are in the same kind of problem that the RNA at the beginning, saying, you can compare across sample, just don't believe a lot what you have intrasample, the difference in there. But for normalization right now, I mean, I'm using DSEC2, the only thing that is doing is normalizing as the RNA-seq. And I mean, it's kind of simpler in the sense that you don't have the length problem because you are always getting the same sequence all the time because it's super short. But nothing else much to do.
So I'm going to throw out a bomb here that's going to
that's good. Bombs
change
are good.
everything that you guys are probably change everything. I mean, probably shouldn't. The paper, it will be useless, but possibly QIAGEN has a brand new kit.
Okay.
I'm normally we're just finishing up our first round of experiments with it, but it's done the right way. So it's got UMIs.
Okay.
So you get like every ligation event will have a unique molecular index.
Okay.
So all the PCR effects go away. Where they're claiming, and I haven't seen the data on this, and I've tried to get my hands on the reagent, but they weren't willing to give it to me. There is a reagent out there that's your test reagent for bias, and that is the synthetic library of all of the different microRNAs. It's a very, you can't make it yourself. It costs way too much money. Apparently it does exist and it floats around. And like I said, I asked for an aliquot and they wouldn't give it to me. But their claim, so the initial results from the first paper about bias showed about 5 orders of magnitude variation using this reagent, with very little in that kind of twofold range. The BioKit expands what's in that twofold range about twice, but there's still a 5 order of magnitude.
Yeah.
QIAGEN's claim is that everything is within that twofold range, which would be amazing if it is.
Yeah.
I don't have the data to say this.
Okay.
But if it is true, that will change.
Oh, that
But
would be
like I said,
awesome.
but more important, the UMI is removable. All of the PCR artifacts. And so now you're just, what type of microRNA is it and how many UMIs do I have? And so, I mean, all of these missense things, I can't tell if they're sequencing errors or not, right? I mean, in theory, they could be, unless somebody's taken them and mass spec them to prove them. I don't know the literature well enough to know if they've been validated by mass spec or something. How do you know they're not a sequencing error?
But I can understand the sequences about the mutation, maybe addition, but the one that are trimmings. So.
Oh no, the trimmings,
okay.
but those flavors. But at least they'll have the same UI.
Yeah, that's for sure. And the addition has been validated, like some of them, for sure for nucleotide mutation.
Right, but now you'll have UIDs that are
so
really, and that is absolute, right?
Yeah.
That's there no matter what.
Yeah.
If there, you can't look it up, it's not out yet. If you're going to try.
Well,
we're late data, if you will. I'm not under any NDA on it. They didn't make me sign any paperwork, so I'm not under NDA on it. But it's, I've seen lots of kits come and go. And, like bio, okay, that sounds like it's better, this, that, the other. This is one I was like, okay, I'm going to find a project so that we can get some data on this because I really like a lot of what they've done. The downside is it will not do small RNAs. It's very targeted for microRNAs. There's some weird chemistry in there.
Okay.
And it may be that it has things that drop out. Now, again, I don't know. It's hard for me to know without this reagent that I don't have my hands on. But we put it on some real samples, so we'll know.
Yeah. So hopefully next year we have more information.
I'm hoping next week I can have more.
Okay.
This is on the sequencer right now.
Oh,
This is not theoretical.
So let me know. I mean, I'm super interested to know if there is someone trying to fix that because I think that for some reason, maybe it's because it's more difficult than other things or whatever, but has been stuck in the Illumina protocol like all the time. And I think that
we use NED, which is the same thing as but it's bio is an improvement in theory. I'm really enthusiastic, and I'm sorry. Next year is only two weeks away. So
so it's not that bad. I mean,
I don't think you'll have to wait 12 months.
No. Yeah, I was confident next year maybe I don't know, like 1 month or two, just you play with the data.
Well, we'll see. I mean, like I said, I want to know the answers soon because I'm really enthusiastic about if this works. I mean, it requires a paired end run instead of a single end run, just like all the UIDs do. But it really, I think, has the possibility of really. I've discouraged people from doing microRNA sequencing because I don't think I'm worried about what it does, which was the absolute counts. And I was involved in some of the earliest papers on microRNA sequencing.
Yeah, I mean, for sure. I mean, I don't trust like many of them is like what is top express? It's like I don't know. But I have to say in terms of differential expression, there is a good rate of validation with if you trust PCR as well at the same. And then some of them have been done even functional analysis. So I think that, I mean, it's nicer than any other RNA-seq or any other, so I agree on that. I think that in the first expression, if
But it's like ChIP-seq, where there used to be ChIP-seq, where there used to be algorithms to try and interpret between the points. Now ChIP-seq, so it doesn't matter anymore.
Yeah.
So a lot of the old algorithms for microRNA sequencing, maybe you're going to have to do some pretty big adaptation of those to make UIDs and things like that work. You won't have to do some of the, hopefully by getting better data, you won't have to do as much gymnastics on the informatics side.
No, that's for sure. I mean, always is better to improve what comes from the experiment than anything else. Totally agree on that. Also, that's awesome. Thank you for doing that. It would be great if we can have something that can give us better estimation.
I think so too.
Nice. Okay, so I think we are all good and ready to go to the call again. Okay, thank you. Bye.